Alright, listen, I know I'm a little late. Maybe a lot later than I should be. It's like almost 10, I get it. Let's just jump through this as quickly as we possibly can, okay? I have had 2 midterms today. Yesterday, I mean to say. YouTube hasn't cooperating. OBS hasn't been cooperating. Let's just get this video over with, okay? Here's the disclaimer. Here's transcription worksheet, okay? Question 1. Does not proofread. So basically, the conclusion is the RNA application is not better, okay? This simply means it makes more errors, okay? So what I mean by this, it makes more errors because, well, this isn't really a solid theory. This is just a general point it came up with, but not even scientists can debunk this because it's not a real theory. But there's no corrections that are going to be made by the proteins for the proteins to correct this. What's going to be the significant? A lot more errors for RNA than in DNA, okay? So for transcription, start-stop signals, basically, this is the start signal for, for prokaryotes. These are the two needed, and then these are the stop signals that are absolutely needed. Row dependent is its own thing, and intrinsic is basically you meet a stop signal, drop it. Okay, these are the stop conditions for the eukaryotes. Eukaryotes are very tricky and confusing, but this is mainly because of evolutionary purposes, and it's found flaws, and it's attempted to fix those flaws. We're still trying to figure out how these flaws are being fixed, but it is what it is, okay? So a good acronym to remember, just these guys alone, much less Tata box and Cat box and GC box or GG box. T5DAPFA, good acronym, okay? And then how to stop the eukaryotes, the addition of the 5 cap and 3 polycap, CDT. And quick disclaimer, to this day, we don't know the complete, or through the education so far that I've taken, it's still not complete. We don't know how it ends it. We just know that these are primary components. This is more specifically for RNA, but CDT for sure. We'll get into more detail about CDT later. Let's just go to question 3. I got lazy. I got fucking lazy, okay? So box, the yellow highlighters. You see it right here? You see it right here. I just highlighted it, okay? Highlight and label the template strand. Boom. 5-3 end. You see it. Okay, listen, I didn't do the opposite end because I'm a lazy bitch, okay? That's what it is. I'm just a lazy bitch. But you can probably get 5 end, 3 end, 5 end, 3 end, okay? It's that simple. Transcribe the template sequence below. If you're happy and you know it, clap your hands. Alright, now let's just move on. Basically, you just figure out which one is the template strand and then you do the coding. That's it right there, okay? So consider and determine the non-template strand which it is coded from. Ta-da. You're wondering where I got this? From right here. I don't know how they did this. It's so annoying. So I just made my own thing. And indicate 5 and 3 and for this. Okay, just ignore this. I just did it right here. I made a mistake earlier. Just from a prior recording that I couldn't upload, but forget it. And this is the arrow in which it's being transcribed in from the DNA, okay? Because this is the 5 end, this is the 3 end. And if it's true what it's saying about the thing, it's basically, it's attaching the 3 end, and the 5 end is just being extended out because it starts with the 5 end, and it's just being extended out like it's a, I don't know, knitting. Yeah, knitting is the best example I could think of. Okay, is it possible for prokaryote eukaryotes? No, just simply no. In conclusion, no. Prokaryotes can do multiple, but eukaryotes can only do one at a time. And this is the more technical reason. You can pause and read it. You can just pause and read it. Yeah. Okay, question 6. Copy paste from the frickin' lecture notes. I think it's lecture 9. Good luck finding it. I'm not gonna remember this exact slide number. I'm not that specific. Okay, sigma factor. Okay, there's no accurate region or parameter. Okay, this is like no sigma factor, okay? So if there was no sigma factor, this is what I argued. This is what I would argue again for row proteins, transcription and termination. No catalyst, because that's what RNA polymerase 2 specifically focuses on. RNA polymerase 1 and 3. I threw up in my own mouth just saying this. Disgusting, I know, but I hate biology. Especially this level of detail. We should have technically stopped here, but it's 20 questions long. I'm pretty sure you're all knowing. Okay, this is the table. Copy pasted from the lecture. I don't know which lecture this is from, but good luck finding it. I wouldn't encourage doing it from this video, but that's your risk. Okay, so what are the advantages and disadvantages for splicing? So for alternative splicing, I'll explain that later, but I'll first explain RNA splicing first. But basically, RNA splicing was once you produce the RNA, you have pre-mRNA, okay? So basically, with this pre-mRNA, it has extrons and it has introns. You already know what that is. But what the eukaryotes desire is the extrons only, because that's what's going to be specifically coded for. The whatever the thing is, spliceosomes, it recognizes this and it wants to remove this. It's going to be removed with sRNA, snRNA, but that's just me speaking ahead of time because of MCAT prep. Thank you, MCAT. Okay, but for real though, takes pre-RNA, cuts out all the introns, just gets rid of it, and then extrons only all attached to. That's what RNA splicing is. Alternative splicing is basically an argument that we make multiple proteins from one gene. How's this possible? Basically, mRNA, whether it has mistakes or not, okay, it's meant to specifically translate into one protein, okay? But let's just say we want to make multiple other proteins with similar functions and shapes. What the body or what the evolution process has decided to say is, in order to save time and energy, and we found a more effective shortcut, we can use one pre-RNA, which we know we can produce multiple shapes and multiple multiple different proteins, similar in shape and function, may have a greater effect, may have a lesser effect, but it gets the job done. We can do it with one pre-RNA instead of going through the whole process of transcribing, translating from DNA to RNA. You see where I'm going with this, okay? What are the disadvantages? By the way, these are both advantages. Disadvantages, mutations. This can call alteration to the reading frame, both for DNA, both for RNA. And changing the structure intended for protein and can create a premature stop. No one likes a premature stop, okay? No one likes a premature anything, okay? It doesn't make the right transcribed mRNA. That's all it is, okay? 10, 12, 11, 15, all opinion pieces. If you don't agree with it, let me know in the description or comment section below. Please do so anyway. DNA, RNA, polymerase, incorrect protein production. This is basically what I would argue, basically. What's the result of not doing it right? Okay, the opinion piece for this is that I would say extrons over introns because one, they're the ones being expressed, and two, they're never removed. You could say introns because they can introduce new variations, new coding, whatever, but basically since the introns are being removed and destroyed, it could leave room for experimentation. I'm not denying that, nor am I arguing it, but I think extrons have more power because if something happens one way and the body decides it's more efficient, it's effective, and it's better, it's going to keep doing it that way. Basically 12, I highlighted the important thing here. Gene was useless because it contained too many amino acids. But basically, as we were arguing for RNA splicing, too many introns attached to the protein, likely making it ineffective because it was too long. Basically, just self-justifying it. Okay, CDT is new. I had no idea what this is, so I posted everything I know about this here. CDT is a long terminal domain. Basically, it's just a long carbon chain, but it's very confusing and unique. But basically, this helps for transcription and elongation for termination. Without it, like the removal of CDT, it inhibits all hnRNA processing. Okay? Heterogeneous. What the fuck? Oh, it's just pre-mRNA. Are you kidding me? It's just pre-R. Okay, you know what? Just forget it. Okay, the phosphorus of CDT performs modification of hRNA required. Okay, so basically what this does is it phosphorylates the CDT for the close modifications for the pre-mRNA in order to be made into the mRNA. Okay, so basically you can see here that when the raw mRNA is made, like straight from transcriptase, okay, it's just fresh out. It's just been cut. It's been made. It's pre-mRNA, and then it somehow becomes hRNA. I'm not too sure how. CDT comes in at the opportune time and fixes it into mRNA by its phosphorylation and modifications to the hRNA. So this is where I would argue CDT does the majority of its job. Basically, if you were to describe how it functions, it's like a docking site right here and extends the phosphorylation to which various other proteins which we don't know about but we'll learn in the future, can dock in and make modifications to the RNA. If you had to take anything away from it, it's a capping complex, binding protein, and recruits lysosome. That is its main perfect function. Okay, so length of poly A tail. How much time do I have? Whoops, leaked my DMs. Shit. Okay, so there, for the length of the poly A tail, it's 20 bases long. Well, the message is cleaved around 20 lengths long. So there's a poly A. I'm gonna have to cut this out. I'm gonna have to merge it. Fuck this. Please pretend like you never saw anything, but moving on. Yeah, basically there's a 5 cap for pre-mRNA, but the paper doesn't discuss too much about this, but this is also integral for making pre-mRNA to mRNA, like mature RNA. So basically this is the termination of transcription, like dead end. Like once you do this, it ends. Like once you add the polytail, boom, over. Okay, so 15, like I said before, was an opinion piece. Basically, I just left it as if the DNA transcription like signals don't or triggers don't work anymore, what could happen? You can obviously say like it doesn't do its function that was intendedly built for, or it could be a waste of time and resources. That's such a cruel way of putting it, but it could have negative consequences. That's essentially what the main argument is. Okay, now 16 through 20 basically goes over these images, and be sure to check out Smart Biologies, more specifically 5.1, 5.2, and there was another one for row protein, but I don't remember that one. But 5.11, 5.12, go through those. Okay, for 16, basically the blue mutant sigma MILF. That's basically what I said. That's what it's called. The blue subunit is the sigma. This right here, that's what it is. And what are the sequences? These are the sequences right here. I'm basically I just got bored and just started posting memes. All right, give me a break. Okay, so basically okay, fuck that. Just redraw figure 2 and just label everything. Okay, redraw, give the arrows, just draw everything. Okay, I could care less. Okay, figure 18 was question 18 was about figure 2 and figure 3. All right, these two. I'm sorry. I'm trying to make this work somehow. Okay, so figure 2, basically it was like play spot the difference, like find Waldo, okay? The one is that one has the sigma factor and the other doesn't. You could talk about the consequences for this, such as because of the sigma factor, it could be in the way of producing the mRNA. Basically, it has to detach in like 10 subunits or so. And are they still at the promoter site? As you can see here, that red, not there. And then you can see here, they're still at the promoter site. That's what I would say, but you could make more of an argument. 19, okay, I'm gonna be completely honest here. I have no idea what 19 was talking about. I saw the video, I still didn't get it, okay? But basically what the main argument is, what I thought was, there was no proximal elements control. Basically, can you think of an example where it's non-self-complementary? There is an example, which is the proximal control elements, okay? So basically, no, it's not all of them. Not all are self-complementary, and it makes transcriptions before proofs. Okay, take this with a grain of salt, basically. But yeah. And then lastly, question 20 is about needs to be open up for DNA RNAs. As we said before, termination of transcription. Remember this from way earlier, like question 3. That's where it is. Now, there is a whole section below where it's like, do this. I'll link it in the description. Okay, listen, I know I didn't do it for lab 2. I completely forgot. It slipped my mind. Fuck that shit. But anyway, like any antisocial reject child who has social problems, I'm gonna say this. Please like, subscribe, and share this in the description below. Or with your friends' family, not in the description below. You can find the link in the description below, but do whatever you want. This is my help to you. Bye.